Good afternoon, everybody, and a very warm welcome to our event, Science for Sight, Shaping the Future of Eye Health. Before I introduce myself, I'd like to start by acknowledging the traditional custodians of the land, the Gadigal people of the Aurora Nation, and pay my respects to elders past, present and emerging, and extend that respect to any Aboriginal or Torres Strait Islanders here with us today. Just a couple of housekeeping points before we get started. In the unlikely event of an emergency, we'd like you to evacuate through these doors straight in front of me and out into the main entrance on Missenden Road. The bathrooms are just, again, out these front doors and on the right. And just to let you know that we will be recording this session so we can share it with those who can't be with us in person today. So my name is Dr. Jessamy Tiffin. I'm the Deputy Director of the Centenary Institute, and we are a world-leading medical research institute. We study a large number of diseases, including cardiovascular disease, cancer, rare diseases, infection and immunity, healthy aging. And our major goal is to understand how these diseases work so that we can try and come up with better treatments and get better outcomes for our patients. But of course today it's all about eye health. And I think like many of us who have healthy vision, it's probably something we often take for granted. But if you could just imagine losing your ability to read, to clearly see the faces of your loved ones, to just be able to confidently and independently navigate your way through your day, it can have a huge impact on people's lives, and this is where research becomes vitally important. So just to run through what you can expect today, we're going to be hearing from a leading clinical researcher, two of our amazing eye researchers from the Centenary Institute. We're going to be having a panel discussion, and we're going to be hearing from two very special guests that they're going to share their experiences of what it's like to live with some of these eye disorders. There'll be an opportunity for some questions and answers after that. And afterwards, we're hoping that you can join us next door in the restaurant so you can continue discussions with our speakers over some refreshments. So first up, I would like to introduce our first speaker, Professor Stephanie Watson. So she's the head of ophthalmology at the University of Sydney. She's a leading eye surgeon and researcher in cataract, corneal and laser surgery, and an internationally recognised innovator with over 200 publications. She's the chair of the Ophthalmic Research Institute of Australia, an advisor to the Stem Cell Therapy Mission. She's also the recipient of major awards such as the New South Wales Premier's Prize for Leadership and Innovation. So please welcome Stephanie.
Okay, thank you. Thank you. Thank you for those kind words of introduction, and thank you all for coming today. I'm going to talk about some of the eye conditions that we've been researching on, and then following me, Professor Lyons will talk about some of the research aspects. So first of all, worldwide, there's about 2.2 billion visually impaired people, and this is causing both direct and indirect costs of over 620 billion. So it's a big problem. And there's hope that these cells, stem cells, may one day be able to alleviate some of this burden. Now, stem cells, for those that don't know much about it, they're actually the building blocks of the body. So from a stem cell, you can make other cells. You can either make the same cell as that stem cell. So if it's a skin stem cell, it could make skin. But there's now also stem cells that can be programmed to make other tissue types. Now the cornea is the eye's window, and it's composed of a number of layers. The very top layer is a layer of epithelium, and this is like a covering layer, and it prevents the cornea from getting infections and from other diseases and from trauma. Now, how stem cells work in the cornea, this is actually a diagram to show where they are. They actually live at this junction zone. This is the cornea here, which is clear but looks coloured because you're looking at the iris, and this is the conjunctiva. And at this zone between the cornea and the conjunctiva is where the stem cells are living. And they're in these structures called palisades of Vogue, which are vertical sort of little clefts. They're a bit hard to make out. And they're in folds because we think that protects the stem cells from the environment. And how it works is the stem cells sit in these folds at the limbus and they divide. And when they divide, they make another stem cell, but they also make an epithelial cell that goes across the cornea. And every 7 to 10 days, this epithelium is completely replaced. So these stem cells are very busy pumping out these new cells. And it's a bit like a garden, where the stem cells are the seeds, and then they're producing the flowers, which is the epithelium. But for our garden to be healthy, we also need to make sure there's no weeds, there's enough rainfall. We developed a model to try to understand more about stem cells and how they behave on the cornea. Because if we can get the stem cells to do what we want, we can then repair corneas. We can repair the epithelial surface, which will help prevent infection and improve vision and prevent scarring. So this is our model here. And how stem cells actually move across the cornea is in whorls. And so each colour here is a different stem cell colony. So this is the limbus around here, and the stem cells are there dividing away. And as they divide, they move across the cornea in these whorls. And interestingly, it's probably a bit hard to tell unless you're an eye specialist, but on looking at on patients' eyes when they develop failure of their stem cells, they get this whorl-like pattern because of the way the stem cells are moving across the eye. Okay, so this is a bit like my garden, this photo here. There's lots of dead and dying plants. And unfortunately for some patients, they have deficiencies or issues with their stem cells, which means the front of the eye is not healthy. They're more prone to ulceration, which is painful, reduces vision, and also predisposes to infection. There's this condition where the stem cells don't work is called Limbal Stem Cell Deficiency. It's more common after chemical injuries, after trauma. Some people can be born with a genetic problem that means that their stem cells don't work properly. And the end result, this is a patient of mine who, when he was 23, had an injury from beer line cleaner. And in order for beer to taste good, the beer lines are flushed with sodium hydroxide at very high strength. And that unfortunately, the mechanism linking the tubes together was loose, and the sodium hydroxide got splashed into his eye at force. And this was the good eye. The other eye completely dissolved. And so with the lack of stem cells, he's developed this severe scarring and vascularization. And what we're trying to do is produce treatments that actually can help him to see again. He got married, he's had 3 kids, but he hasn't seen his children yet. And we can't treat it with standard grafting. Another condition we're looking at is a condition that affects the middle layers of the cornea. So if we look at our cornea, it's a bit like a sandwich, and you've got this sort of cell surface on the top here, but most of it is actually layers of collagen. So about 90% of the cornea is collagen, and it's quite tough. It's a bit like sort of a piece of steak or tendon in consistency, but it's clear because the collagen is arranged so precisely that it's transparent, that light cancels itself out. So in this condition called keratoconus, what happens is these layers of collagen slide on each other. And so the cornea starts to change shape. It's a bit like a sandwich that needs a few toothpicks in it to put it back together. And patients with keratoconus, this is a simulation of what they might see. They get a lot of distortion in their vision because the cornea is the lens of the eye. And when it's not a normal shape, it's a distorted shape, it distorts what they're seeing. And the condition is progressive, typically onsets in young adults or children, and progresses for a period of time. And it basically results in the cornea being protruding and being misshapen. This is a scan from a patient, and what it is it's looking at, it's like a geography map. And so the shape of the cornea, if you look here, you can see it's green, there's a little bit of a cross pattern, and that's a little bit of astigmatism where the eye's not round. So it's a geography map where there's a little bit of steepness, but the patient went on to develop keratoconus, and you can see here that now there's a big red area in the centre, which is an area of where the cornea is steep. This was during COVID that they came back and had progressed. So lastly, I'd just like to talk about dry eye. Now, this is a diagram showing what the front of the eye is like. We call it the ocular surface. And it's a very complex system that actually produces our tears and keeps the front of our eye healthy. So we have a lacrimal gland that produces the watery part of the tears. But in the eyelids, we have these meibomian glands that produce the oily part of the tears. And you need the oil because water is like just putting your hand under a tap. So the tears need to have more than just the watery part. We then need to make sure we've got blinking, so the nervous system active. And we need lids in the right position in order to stop the tears evaporating away. The tears then drain down into the nose here. There's also immune factors. One thing that happens in dry eye is people get a lot of inflammation on their eye. So this is what you might be looking at, but if you have dry eye, then your vision can be blurred. And this is because the tear film is the final lens of the eye. It's what you look through to see clearly. And when there's a tear film that has dry eye, it's like a car windscreen with rain on it. You've got blobs and it breaks away and you can't always focus clearly. This is looking at how we can assess dry eye. So how we see it is we see this is like some rings that have been projected onto the tear film, and here a patient with dry eye, you can see the rings are kind of breaking away. And this is a dye that we use called fluorescein that actually shows the cells that aren't healthy due to dry eye. So corneal blindness, all up, it's a common cause of blindness, and unlike macular degeneration and glaucoma, it really affects all ages. For blind children, they've found that if children suffer with corneal diseases at a young age, they actually have a lifetime of morbidity ahead of them. And it's been noted by the WHO, World Health Organization, that corneal scarring is actually a priority disease because of the burden it inflicts on patients. Thank you. So I would now like to introduce Professor Guy Lyons, who's a leading scientist at the Centenary Institute and has a long and distinguished career of investigating in both the skin and the eye, trying to find the causes so we can find the cures. Thank you.
Oh, thanks, Stephanie. My lab at Centenary works on two of the diseases that Stephanie mentioned with Stephanie. I went the wrong way. So we're looking at keratoconus and limbal stem cell deficiency. And the things you need to be aware of for our work, the key things are that apart from focusing the light into the back of the eye, the cornea provides protection from the inner structures, the lens and the retina and so on. And indeed, not just from germs and from chemicals, but from ultraviolet light as well. As Stephanie mentioned, the outermost layer of protection for the cornea, the epithelial cells, are derived from stem cells located in the limbus at the edge of the cornea. And it's these cells that go haywire and give rise to limbal stem cell deficiency. So So keratoconus is, as Stephanie just explained, a thinning of the cornea which we believe may be contributed to by ultraviolet light from the sun. So it affects about 1 in 84 Australians, and it tends to cluster in families. So we know it has a genetic basis, but it's a complex genetic basis. By that I mean it's contributed to by several interacting genes, and this makes it difficult to predict who is going to get it, even who in a family that already has a family member that suffers from it. Like many genetic diseases, it's an interplay between the genes and the environment that ultimately determines whether you'll get the disease. And this study by Gordon Shark et al. Found what's called a latitude gradient of keratoconus incidence. So basically, the closer you live to the equator, the more likely you are to get the disease. So that brings us to the question. There are other possible explanations for this latitude gradient, but we're posing the question, does UV from sunlight actually contribute to keratoconus? And because it's the epithelial cells that bear the brunt of the UV, they actually absorb it and stop a lot of it from getting through to the inner layers, which actually show the symptoms of keratoconus. We postulate that it's actually the epithelial cells that initiate the whole keratoconus process. So to determine whether sunlight exposure or UV exposure has a role in driving keratoconus, we obviously need to find out how much UV people are exposed to. And rather than relying on people's memories and writing down what they thought they were exposed to 10 years ago, I know I couldn't do it for the last month, we decided to take a DNA sequencing approach. So what we do is to take tiny bits of tissue from eyes or sun-exposed parts of the body and subject it to DNA sequencing. Now, when the body is exposed to UV, the UV causes specific mutations in the DNA, and when we sequence it, we can see these so-called signatures, which are directly caused by ultraviolet light. And by counting the number of signatures in a person's DNA sequence, we can get a measure of how much light they actually have been exposed to, an objective, reliable measure. And by correlating that with incidence and age of onset of keratoconus, we can see if there's really a direct association there. To determine whether the epithelial cells are driving the keratoconus process, we're using our preclinical models, and after a low dose of exposure to ultraviolet radiation, we can look at the corneas and see what changes they have and if they resemble to keratoconus. So these are one of the eyes that Stephanie showed before, where each of these clusters of cells arises from a single stem cell located in the limbus. And we have other models where the cells are outlined by a fluorescent marker, so we can actually watch the cells as they grow and move around in intact corneas to determine the effects of UV on their growth and turnover. And we also have a computer simulation model which enables us to plan our biological experiments and interpret them. So we hope the outcomes of these keratoconus studies will be to provide evidence that will enable us to drive a public awareness campaign, particularly to keratoconus families, to ensure that they can reduce or at least mitigate the effects of exposure to sunlight. And secondly, to give us the information needed to find drugs that enables to block the pro-keratoconus effects of sunlight and so enable people to get the beneficial effects of going outside. So the second disease is limbal epithelial stem cell deficiency. As we heard, it results when the limbal stem cells don't function properly, either because they're damaged or because of faulty genes. And you get the overgrowth by the adjacent conjunctiva, which interferes with clear vision. At the moment, the main therapy for this is transplantation of limbal stem cells. And this is a two-step procedure where the cells are obtained, usually from a donor, and then grafted surgically onto the affected eye. So transplants have issues. Usually they're from donors and quite often cadavers, and quite simply there aren't enough of them for the number of people who require the transplants. The other main issue with getting transplanted tissue from other people is that the patients require to be on immunosuppressants to stop rejection of the transplanted tissue. Sometimes this doesn't work and the tissue is rejected anyway, and when it works it needs to be maintained, which leaves the patients susceptible to infections because their immune system is reduced. In cases where patients have one good eye, they can get tissue transplanted from that eye to the affected eye, but this runs the risk of damaging the good eye, the one good eye, and it's also not a solution for genetic conditions because both eyes are affected. So we want to redesign the limbal stem cell deficiency transplantation procedure to look something like this. The patient goes to a clinic and gives some blood, and then goes off and does their own thing for a few weeks, and then goes to an eye surgeon like Stephanie, who administers a contact lens which is filled with these stem cells, and they get put onto the patient's eye. Now there's still some preparation of the eye required here, but there's no immunosuppression involved and there's no complex surgery involved. And we would expect the procedure to be have fewer complications and provide less discomfort, and there certainly would be less clinical time for the patient. So our pipeline for developing this looks like this. So we take white blood cells, treat them with certain factors and convert them into these pluripotent stem cells that Stephanie referred to, which can give rise to potentially any tissue in the body. And if we treat those with the right factors, we produce the limbal epithelial stem cells which the patient needs. These are transferred onto specially coated therapeutic contact lenses, which are then transplanted onto the patient's eye. So this is an image of cells that are being converted from our pluripotent stem cells into the limbal epithelial stem cells that we want, and this field shows that they're producing all the markers that they should if they're actually limbal epithelial stem cells. We're collaborating with investigators at the CSIRO to optimise the transfer of our stem cells using contact lenses. It's not commonly known in Australia, but it should be. It's the soft breathable contact lenses that so many of us use today were actually invented by CSIRO scientists in the 90s. So our colleagues at CSIRO have put these fancy coatings on them to see if they would help the cells to grow, and indeed one of them seems to cause a significant improvement in growth of our limbal stem cells. And the cells, by electron microscopy, appear to have all the features that they should retain. So the next step will be to put our iPS-derived limbal stem cells onto the contact lenses, make sure that they grow and transfer properly, and do some safety testing before going into clinical trials. And I'd like to finish up by thanking my colleagues and collaborators, various funding bodies who have supported the lab, as well as Centenary Institute donors. So thank you for your attention.
Thank you. Okay, can everybody hear me? So excellent. We're going to have some discussion now, and I think this is, that's much better. Feel less blinded by the light, so to speak. So we're going to hear some interesting stories that hopefully will help you gain an appreciation for the impact that eye diseases and corneal diseases have, and also the potential for research in the area. So perhaps we'll start with Majde. We'll start with the introductions for everyone. So would you like to go first?
Hi everyone, I'm Dr. Majde Abbasi. I'm a postdoctoral research fellow at the University of Sydney. And I'm working as a researcher on eye disease since 2016.
Kiara?
Hi, my name is Kiara. I'm 21 years old. For the past 13 years, I've been having dry eye syndrome, and I've been a patient of Professor Watson since I was about 10.
Hi, my name's Melissa. I'm 31 years old, and I've had keratoconus for the past 10 years, and have also been a patient of Stephanie's throughout that time.
Settles down. Majde, so what do you find to be the most rewarding or exciting aspect of your job as a researcher?
Oh, thank you. Well, I think the most rewarding thing in our job is that we know that someday what we are doing in lab actually make differences in the patient's health, As in our research, cornea is known as the window of the eye. So anything happen to cornea and damages the eye, it will affect affect the vision. So when we make discoveries in lab, either it is, for example, understanding the mechanism of the disease, or for example, a new therapy for repairing the cornea, it's something like that. It's very rewarding because we think that we are finding new ways of treatment. So it's very motivating for us. The other thing I think, because as a postdoc, I mentor students and I have collaborations with other scientists, it's always motivating for me because I try to show students how impactful is that your research matters for the patients. And what I think the biggest reward is that what we are doing now sometimes, in future, help the patients to gain their vision, which is quite precious.
Thank you. And Kiara, perhaps if you could tell us a little bit, about your dry eye syndrome. It's a very severe type of dry eye syndrome, and how it sort of impacted your ability to sort of work or live independently.
Yeah, before my dry eyes kind of impacted my life, I was able to live a fairly normal life. I was able to study, work, drive, live quite independently, but with dry eye syndrome, like Professor Watson said, you do get a lot of inflammation, and I had quite a bad flare-up of inflammation, which just made my dry eye syndrome a lot more severe, and I wasn't able to drive, study. I had to stop working, and I haven't been able to for the past 18 or so months. I was stripped away from a lot of my independence. I became very reliant on my parents for the very simple things, just to be able to get out of bed, make my bed, prepare meals, brush my teeth, because my vision had suddenly become so blurry, and my eyes, I couldn't really keep them both open at the same time, and I couldn't really find relief in a lot of things, and I just kind of had to learn to adapt to my new life, and just, I don't know, I think it was very difficult. I wasn't able to see my friends go out, and I was just kind of there in my bedroom a lot of the time. So yeah, I don't think I really would have thought it would have affected my eyes that much, but yeah.
No, well, you've been very brave.
Thank you.
And Melissa, perhaps if you could walk us through your journey.
Yeah, absolutely. So I first noticed some changes to my vision around about 10 years ago. particularly while I was studying for some exams. I was really struggling with the computer. I couldn't see my notes, and it was becoming a bit problematic. Previously as a child, I had worn glasses before, and my first port of call was to go back to the ophthalmologist who had seen as a child. His first sort of recommendation was to just give me the prescription that I had last had as a child. And unfortunately, although everything was much bigger in those glasses, it still wasn't any clearer. So we were playing around with some different prescriptions for a while, still couldn't find anything that was addressing the problem. And then, then we started running a whole range of different tests to see what was going on. One of the things they were looking at was there a brain tumour there? Thankfully, that was not the case. But it was quite a sort of distressing period where we couldn't work out what was going on. Eventually, he told me to go and see one of his colleagues who worked at an optometrist that had some different technology that he didn't have. And it was at that appointment that keratoconus was first raised as a possibility, although no diagnosis was able to be found yet. So I went back to the ophthalmologist, and once we'd had the discussion that keratoconus was a possibility, he said, okay, you have to go and see Professor Watson. He was absolutely right, and thankfully we were able to get the diagnosis, and I've been under your care since then, and for both of my surgeries as well. Also quite lucky to have Margaret Lamb as my optometrist, and she keeps me in my glasses and my contact lenses.
Yeah, that's very, it would have been very distressing at the time, being told you might have a brain tumour.
Absolutely.
Yes. So, Moche, looking ahead in the next 5 to 10 years, what scientific developments do you expect to shape the future of eye research and treatment?
Well, in every science we can see that it's moving in a pace that we have never seen before. I think in terms of our research, corneal disease, stem cell therapy is quite advanced now. So I suppose stem cell therapy will definitely help for lots of diseases. Also, I think a combination of stem cell therapy, regenerative medicine, and genetic editing will help to actually correct some of the genetical defects that is not available now. But if I want to actually have a generally speaking, I think something that accelerates science is now artificial intelligence. So we can see that AI now can go through a huge amount of data quite quickly. For example, the medical images of the patients, or for example, genetic data, or patient's history, which if he wants to do it, the human wants to do it, it will take years. So I think AI is also in lab. It's in terms of clinical, but in lab, really AI help us now to design our experiment quite easier, better experiments, better designing our data and analysis. So, but yeah, I think we are moving so fast, and we will have quickly we will go towards something like, this gives me hope that we can have somehow restoring the sight in near future rather than just managing vision loss. Yeah.
Thank you. And it might be interesting to hear from Kara at this stage about how your condition has shaped your outlook on emerging treatments in research, and what do you want researchers to deliver for you?
I think my outlook is sometimes it can be quite scary. I think with having Stevens-Johnson syndrome and dry eye syndrome, the research out there is not quite consistent because there isn't enough cases, and I think that the options that are currently there are quite limiting, and it's always exciting, I think, when there is something new, but I think at the same time, with Stevens-Johnson's and dry eyes being so rare, it can be very daunting to want to try something new, because I think you don't want to be too hopeful because there's not really a backup option if it doesn't really work. And I think even though it is medically approved and it's safe to use, I think it can still be very scary because there isn't too many well-known cases and you don't know what's really going to happen. But I think at the same time, with that being said, there's been so many changes to medication from the time that I got sick to now. And I think that without my current treatments, I wouldn't be able to live and I wouldn't be able to function the way that I am now. And I think technology is always changing, so I don't think it's, I think it's scary, but I think it's good. I think the scientists, the doctors, they know what they're doing, and all I can do is just trust them.
I think you raise a good point about, having a condition that's not common and the need for data.
Absolutely, yeah.
We need those cases. If we could get all the information from everyone around the world, with these uncommon conditions, we'd learn a lot more.
100%, yeah.
And maybe artificial intelligence can help us kind of bring that all together. Yeah. So, Melissa, how has your condition shaped your outlook on emerging treatments or research, especially things like regenerative medicine?
So I definitely think that the idea of having regenerative medicine is, it's an amazing thought. The idea that there could be something that is not just going to provide sort of management, but that could provide improvement and, eventually one day maybe even more than that is quite amazing. And I think I've always had quite a deep respect for medical research. Growing up living with my nan for a long time, she lived with multiple sclerosis, so I did definitely see firsthand how ongoing medical research and treatment can really help someone living with a sort of a chronic condition. But I think definitely that's had a new personal sort of appreciation since going through this experience myself. And I think it just really goes to show that, and something that's really taught me, that the value in medical research is not just in finding those cures, but also in helping people to be able to keep their independence, have that confidence, and be able to have a good quality of life, and just reinforces the importance of investing in those sort of lasting impacts as well.
Yeah, no, that's so true. And I think also, in medical research, it's important to support the careers of the researchers. So, Mojo, do you want to tell us how you became an ocular scientist?
Yeah. So eye has always fascinated me, and I think vision is one of the senses that people value the most. And in my idea, losing vision is the hardest thing that people can go through. Now we know for different type of corneal disease or generally eye disease, treatments are available, but they are not 100% efficient. Therefore, since early my studies, I was always thinking that I want, area research would be my ocular science. So with the earliest steps that I could select my research area, I went for ocular science. So during my PhD, I studied about retina and cornea and how the damage can affect them. For example, disease like glaucoma or aniridia. And during that years, I figured out how complex this field is, but at the same time, it was hopeful because I could see that technology is, progressing so quickly. So that's why it motivated me that I continue my research in my postdoc within the same research area. So generally, I think what kept me in this journey is both the science curiosity and this human side that patients and their families, they are always expecting us, they are waiting for us to find new treatments for their disease.
That's very true. And Chiara, what do you think are sort of the unmet needs for, people that suffer with these severe diseases, particularly from a young age?
I think definitely with current research being a little bit limited with Stevens-Johnson syndrome and severe dry eye syndrome, I feel like there really isn't too much that you are able to do. It's kind of just learning to work within your means, I guess. And I think that eye drops, they're great, but I think that they do provide more short-term relief. They don't really have something out there that can kind of help you a little bit more in your everyday life. And I think there just isn't enough awareness around dry eye syndrome. I think that it's very easily misunderstood. I think people easily just think, you put in eye drops every couple of minutes and that's it. I don't really think people realize how much it impacts your everyday life.
No, that's very true. Look, just to wrap up, so if we could produce a little contact lens with stem cells on it, or there was some sort of regenerative therapy for keratoconus, what would you think? Would you have it?
Yeah, absolutely. I think if there's anything that we could do to have something like that in for keratoconus patients, that would be a really incredible step forward, and just to show how far science has come. And it would, I really hope that something like that could become a reality one day.
We might just finish up by Mojse just telling us about some of the current challenges in science.
Yeah, that's an important question, I think. Science is powerful, but we know all that science have limitations and challenges. I think the most important thing is time. Science takes time. So moving from the research and what we find in lab towards the real world impact, it may take years or even decades. The other thing that is also related to time, I think science is not existing in isolation. So other issues influence science, like for example, social issues, for example, regulatory issues, ethical issues. All this takes time. So if we might find a promising technology in lab, but to make it, widely used, it should be proved to be safe, to be affordable, to, be acceptable by community. So the other, I think, challenges, not only for our research, but all the sciences, is, I think, resources, like funding and technology, new technologies. But I think we shouldn't see these challenges as barrier, I think they are somehow opportunities for us, because somehow it pushes us to have more collaborations, to share our knowledge, to solve our issues, and make more innovations, actually. So.
Excellent. Thank you very much. So if everyone could thank the panelists. Thank you very much.
So if I'll just get you to stay there for 1 second. We now have the opportunity to take any questions from the audience. So we've got Sarah with a microphone roaming around. Would anybody like to ask any questions for our panelists or our researchers?
And don't be shy.
Thank you for a great discussion. This is a question, I think, not for the panellists, but for the doctor. Was it surprising that an ophthalmologist didn't think about keratoconus and was sending somebody to the glasses place, what do you call it? Optometrist, and then when they were sent back to the ophthalmologist? Is it so difficult to identify, or is it specialised equipment, or?
I think early on the signs can be subtle, and so that sort of the cornea changes shape. It's progressive, so early on it may not be obvious, and not everyone has the same equipment. As Professor Lyons was saying, it's been reported, they did a study in Perth where they put 20-year-olds on these scanning machines that can pick it up, and they found 1 in 84 had some signs of keratoconus. So we are getting better at picking up, but we need to have the equipment and also the thought about it, yes.
So I suppose the follow-up question is, should ophthalmologists be made more aware to look for it earlier?
Yes, we do. We also go out and lecture ophthalmologists and point out that we need to pick it up earlier. Because now, before there used to be no treatment if you picked it up early, but now there's a treatment called corneal cross-linking, and so it's really important, for optometrists, ophthalmologists to think about that there actually is a treatment. Because in the past there wasn't a treatment, so some of them may also have that mindset.
Oh, thank you very much.
One in the back.
Part of your literature, or at least the message that came out, said that more women tend to have dry eye syndrome. And I was curious about that because I suffer from that myself. And I wondered why. Is it genetic? Is it to do with, various areas, ethnic areas of the world? Or what have I been doing, Have I been staring at the sun too long?
Look, it's multifactorial. There's different types of dry eye. There's dry eye, associated with autoimmune diseases such as Sjogren's syndrome, and women, unfortunately, have more autoimmune diseases than men. There's been various conjecture about whether it's related to hormones. Some studies say that menopause makes it worse. Other studies don't say that at all. Dry eye is more common generally with age, and women tend to live longer than men. So there's quite a few things, but I don't think you've been doing anything wrong. There's nothing wrong with being born a woman. And
Any other questions from the audience?
This question is for Melissa and Kiara. So regarding getting diagnosed with a rare, uncommon condition, where did you go to get support or more information about your condition? Was there like an organization or body, or did you go towards your clinician for more information about your condition?
So I think early on, once I had the diagnosis, definitely a lot of information came from Professor Watson. There was also a group, Keratoconus Australia, and there was some sessions that I went to when I was early on after my diagnosis to hear sort of other people's stories as well and get some other information about that. And also through my optometrist, Margaret Lamb, who is sort of very leading in the field of keratoconus in the sense of the optometry side of it as well.
I think for me, I find it a little bit harder to find support. I think with the type of dry eyes that I have, I think that there aren't too many other people who have it, and I think that there really isn't a lot of research or known cases of it. And so I think that way it's just a matter of kind of bringing more awareness towards it so that more people understand it better and more people are able to provide that safety and support for other people.
Just another follow-up question for Melissa. So you are currently being cared for by an optometrist and an ophthalmologist. I was just wondering, like, what sort of roles do they play in caring for your condition?
Yes, so certainly in regards to Professor Watson, sort of the ongoing management and monitoring of my vision. I've also had the cross-linking surgery in both eyes. The left one was just done earlier this year, so we're still only a few months post-op on that. So it's much more of, I suppose, from that side of things. From the optometry side, that's where all my glasses and my contact lenses as well, we work through them. I'm actually in the process at the moment of looking at the scleral lens and the new technology that are providing, that's a lot more comfortable. So I'm in the process of getting fitted for those at the moment. And there's also some monitoring that Margaret does as well when I visit her too.
Thank you very much.
Well, if there's no further questions from the audience, that will come to the conclusion of our event. So let's just thank our speakers for their wonderful insights. And I have some goodie bags here that I'm just going to distribute.
Thank you.
I'm assuming this is good for the eyes.
Thank you so much.
And if any of you would like to learn more about the research being done at the Centenary Institute, please scan the QR code. And there's also the opportunity to make a donation towards this wonderful research that we would really very much appreciate. So that concludes the formal part of the event, and if you'd like to, we'd love you to join us next door in the restaurant for refreshments, and you can continue chatting to our speakers. So thank you very much, everybody.